Welcome back to Western Blotting, this time part 3. Please don't forget to check out my blog, where you can find a detailed written protocol of the video content you are about to see. Let's dive right into part 3. So after we have blotted the proteins on the membrane, we have to disassemble our blot. So quite often the membrane sticks to the lid, but this doesn't really matter. You take off the Whatman papers, and what I do, I immediately mark the protein bands of the marker with a red pen. It's very important to do that because if you later take an image, it is actually quite difficult to see the bands unless you mark them. Next, I place the membrane in blocking solution. I let the membrane in the blocking solution for 45 minutes. After the 45 minutes, it is time to apply the first antibody. For that, I simply dilute the antibody in 5 milliliters of blocking solution. I use a special technique in which I place the antibody into a 50 milliliter tube, and then I place the membrane into that tube. This technique allows you to save a lot of antibody. It can be a little tricky to place the membrane into the tube, but with practice you will succeed. I place the membrane on a roll shaker overnight for 16 hours at 4 degrees Celsius. The next day, I take the membrane and I wash it 3 times with TBST. I take a new container, fill it with TBST and place the membrane inside. After that, I wash it 3 times with TBST. Each washing step takes 5 minutes. Now it's time to prepare the secondary antibody. I use a rabbit antibody with horseradish peroxidase. I prepare a dilution of 1 to 15,000, basically 20 milliliters of blocking solution and 1.3 microliters of secondary antibody. After the last washing step, we place the membrane into the secondary antibody. I let the membrane dry a little bit because I don't want to dilute the secondary antibody with TBST. That might still stick to the membrane. I take a new container and place the secondary antibody into the new container. We incubate the membrane for 1 hour and 30 minutes at room temperature. After the incubation is finished, we have to wash again 3 times with TBST. After the washing, it is now time to detect the membrane. Again, I place the membrane on the rim of the container to let it dry a little bit. In the meantime, I set up the software. I select Chemi High Resolution and I take a total image of 10. Next, I place the membrane on plastic. And I add the detecting solution, which I pre-mixed before. You have to mix solution A and solution B in a ratio of 1:1. Then I add the detecting solution drop by drop on the membrane and make sure that the whole membrane is covered. Finally, I add another plastic foil and cover the membrane with it, and I make sure that there are no air bubbles. I place the membrane into the detection machine. Next, I position the gel, or the membrane to be precise. Once it is perfectly aligned, I close it and I run the protocol. And that's it, guys. This is the whole process of Western blotting. I know that it can be quite difficult in the beginning if you are not familiar with the technique, but believe me, with practice you will get these kinds of results, and I really hope that the video helped you, and especially the video series. In total, it is a 3-part video series where I explain the whole Western blotting technique. I thank you so much for watching. I hope you have a wonderful day, and I wish you a lot of success with your experiments. See you in the next video.